I'm Kelly Keene.
And I'm Jackie Ardo. The enzyme we chose to research is glucose-6-phosphate dehydrogenase, also known as G6PD. We chose this enzyme due to its protective nature against malaria.
We will be going over basic information of G6PD, such as function, location, sequencing, structure, and post-translational modification.
We will also be reviewing a primary source about G6PD, the importance of glucose-6-phosphate dehydrogenase activity for cell growth by Wang Ni et al., which suggests the role of G6PD in cell growth.
G6PD is a cytosolic enzyme and is the rate-limiting step of the pentose phosphate pathway, shown here. It provides NADPH, pentoses, and ribo-5-phosphate for the body. G6PD is highlighted in orange. In the pentose phosphate pathway, G6PD catalyzes the conversion of glucose-6-phosphate to 6-phosphoglucotanone, and it provides reduced NADPH here, which regenerates glutathione for the body.
Also, G6PD activity plays a critical role in cell growth by providing NADPH for redox regulation.
This NADPH here is a part of protecting red blood cells against oxidative stress, as it provides glutathione, mentioned before, to the body. Glutathione defends cells against peroxides and reduces these to water and alcohols.
Thought to exist unbound in the cytoplasm, G6PD is more than likely found on an intracellular structure and translocates after growth factor stimulation.
Humans have G6PD in virtually all of their cells, but G6PD can be isolated from a multitude of other species.
A few of these other species seen here are rats, cattle, sheep, rabbits, and horses, because G6PD is pivotal to defending red blood cells from oxidative stress that can lead to hemolytic anemia.
G6PD is commonly isolated from the animals we just listed above to be used in lab settings, including in commercially available kits. This shows to the extent to which G6PD has been studied.
The gene that sequences for G6PD is found on the tip of the X chromosome in position Q28 in humans and contains 14 exons. G6PD was sequenced in 1995 and was originally thought to have come from chromosome 6 and chromosome X.
Here, we see the entire protein sequence of G6PD. We now know it solely comes from chromosome X.
Taking a closer look at the protein sequence for G6PD, the underlying portion of the sequence were amino acids already known from previous research done. The parts not underlined was the completion of the amino acid sequence in 1995 and the realization that the gene for G6PD only came from chromosome X. In total, there are 514 amino acid residues located on the G6PD gene. G6PD consists of 4 subunits, shown in different colors here, each with the beta and alpha domain, and exists in a dimer to tetramer equilibrium. The beta and alpha domain is dominated by 9 curved anti-parallel beta sheets, and hydrogen bonding between these beta sheets contribute to the stability of the enzyme.
There are 4 salt bridges between each of the dimer interfaces between subunits A and B's beta and alpha domains. These salt bridges contribute to the stability of the dimeric enzyme. Additionally, there is a structural NADP+ bound between the beta sheet and the C terminus of the beta and alpha domains of each of the subunits of the enzyme. The NADP+ is shown here, highlighted in red and blue. And here, we can see the beta sheet and C terminus the NADP+ is bound between.
The NADP+ is buried in the enzyme surface, and the binding site has an overwhelmingly positive charge, and has a beta-alpha-beta structural motif. G6PD is post-translationally modified by lysine acetylation, phosphorylation, and ubiquination. Lysine acetylation and phosphorylation are the two most commonly researched forms of post-translational modification.
Lysine acetylation negatively regulates G6PD by inhibiting the formation of active dimers, therefore having a complete loss of activity and sensitizing cells to oxidative stress.
Another form of post-translational modification is phosphorylation, which downregulates G6PD activity. In The Importance of Glucose 6-Phosphate Dehydrogenase Activity for Cell Growth by Wang Ni et al. Suggests that G6PD activity plays a role in cell growth by providing NADPH for redox regulation. Previous research has shown evidence towards long-term activation of growth factors that can be stimulated by G6PD activity. This report was designed to highlight the importance of G6PD activity on cell growth.
G6PD expression was measured by tagging the enzyme with an octopeptide sequence called FLAG. The expression was confirmed using Western blotting and by measuring the enzyme activity in cell lysates. The efficiency of G6PD FLAG expression was measured using immunofluorescence staining using a monoclonal antibody.
Enzyme activity was determined by measuring the conversion from NADP+ to NADPH using fluorescence at 341 nanometers.
Another method used in this paper was Western blotting. It was performed using an SDS-PAGE gel electrophoresis. Proteins were probed with a rabbit anti-rat antibody to G6PD and then visualized using a secondary goat anti-rabbit antibody and horseradish peroxidase reagents.
On the left, we can see the results from Western blotting, showing an increase in G6PD activity expression when growth factors were present.
On the right, we see results from Western blotting when DHEA is present with G6PD activity. Lanes with no bands or lighter bands indicate less expression of G6PD activity due to DHEA inhibiting G6PD versus darker bands.
The results indicated that stimulation of growth and being in the cell cycle increases the activity of G6PD. Overexpression of G6PD was also linked to an increase in cell proliferation.
Dehydroepiandrosterone, also known as DHEA, mentioned before, was also identified as an inhibitor of G6PD. It also inhibited cell proliferation and made these cells more susceptible to oxidative stress due to the lower NADPH levels usually provided by G6PD. Ribose 5-phosphate, also a product of the phosphate pentose pathway, does not rescue these cells from DHEA suppression.
NADPH is also likely a critical pentose phosphate pathway product required for cell growth. Previously published research matches the research done by Wang Ni et al., showing a correlation of G6PD activity with cell proliferation. With the use of previous research, Wang Ni et al. Highlights the importance of G6PD activity that plays a role on cell growth and the usage of optimal cell anchorage. Wang Ni et al. Also points out that NADPH is an important product of the G6PD activity used in cell growth rather than the product of ribose 5-phosphate.
G6PD deficiency is the most common enzyme deficiency in the world. It affects over 400 million people and 1 in 10 African American males in the US. This genetic mutation has a protective effect against malaria parasite, explaining its prevalence. The phenotype is common in men as the gene lays on the X chromosome. The phenotype of G6PD deficiency is characterized by premature breakdown of red blood cells. However, women can still carry the gene, and those carrying the allele may still have the malaria protecting effects from the mutation without the complete presentation of the phenotype. The deficiency is present in various levels of severity, listed as between classes 1 through 5. Most of those with the deficiency fall into class 3, with G6PD activity ranging from 10 to 60% of the normal level.
G6PD deficiency can lead to hemolysis which those with the mutation are exposed to certain medications such as primaquine, which is used to treat malaria, aspirin, or other specific foods such as fava beans, which the other name for the deficiency comes from, favism.
However, usually those with the mutation show no physical symptoms until they come into contact with one of these triggers. Because of this, the deficiency is routinely tested for before any sort of anti-malarial medication is prescribed.
To summarize our presentation, G6PD is a cytosolic enzyme with 4 subunits and is the rate-limiting step in the pentose phosphate pathway. Its location is intracellular bound to a structure and translocated after growth factor stimulation.
Humans, rats, cattle, sheep, rabbits, and horses are all common species with G6PD.
The gene for G6PD is located on the X chromosome in the position Q28, and there are many types of post-translational modifications, such as lysine acetylation and phosphorylation.
Wang Ni et al. Correlated G6PD activity with cell proliferation. Wang Ni et al. Identified DHEA as an inhibitor of G6PD and NADPH as an important product of the pentose phosphate pathway for cell growth rather than ribose 5-phosphate. And lastly, G6PD deficiency is the most common enzyme deficiency in the world, having almost 300,000 deaths being linked to the deficiency per year. These are all our sources we use while researching this enzyme.
Thank you.
Thank you.